Designer babies are a controversial concept that refers to the use of genetic engineering to create children with certain desired traits or characteristics. The term, designer babies, is often used to describe the hypothetical use of genetic engineering to create children with certain characteristics such as intelligence, physical appearance, or athleticism. Currently, there are no technologies that enable the creation of designer babies in the sense of selecting for specific traits or characteristics. However, some techniques, such as preimplantation genetic diagnosis, can be used to identify and select embryos that are not carriers of certain genetic conditions or disorders. Preimplantation genetic diagnosis is a medical procedure that examines the DNA of an embryo before it is implanted in the uterus. PGD is usually performed during in vitro fertilization, which involves fertilizing eggs with sperm in a laboratory and then creating embryos. In this process, PGD is used to identify embryos that are not carriers of certain genetic conditions or disorders. To do this, a single cell is taken from an embryo and tested for the presence of certain genetic mutations or abnormalities. The results of the genetic testing can then be used to select which embryos should be implanted into the uterus. PGD is usually used to prevent the transmission of serious genetic diseases or disorders to a child. For example, it can be used to identify and select embryos that do not carry inherited diseases such as cystic fibrosis or sickle cell anemia. PGD can also be used to test for chromosomal abnormalities such as Down syndrome, which can lead to birth defects or developmental delays. While PGD can be a useful tool to help couples with certain genetic conditions have healthy children, it is a controversial topic and raises ethical concerns about the use of genetic engineering to select for certain traits or characteristics. It is important to carefully weigh the potential risks and benefits of PGD before deciding to undergo the procedure. The use of genetic engineering to create designer babies remains a highly controversial topic, raising ethical concerns and questions about the potential risks and unintended consequences of such techniques. While some argue that genetic engineering could be used to eliminate genetic diseases and improve the lives of children, others argue that it could lead to the creation of a society with genetically enhanced individuals who are perceived as superior to others. Indeed, the notion of designer babies, where appearance and characteristics can be determined in advance, is still widespread among the public. The assumption is that a pair of genetic scissors will make the appropriate changes in the egg and that the baby will be delivered, tailor-made, 9 months later. In reality, however, most of the characteristics we have as humans depend on many different genes and are also influenced by environmental factors. Although it is possible to make certain genetic changes in eggs, embryos, or germline cells, these procedures are still very limited and so far can only prevent or treat certain diseases or disorders caused by defective genes. It is important to note that the characteristics and abilities of a human being are much more complex than just the result of genes and that creating designer babies is still unimaginable at this time. If you don't want to miss further videos about interesting scientific topics, subscribe to our channel and activate the bell to be notified when we publish new short videos. Thank you for watching.